Hello and welcome to the Investigating the Effect of Light Intensity on the Rate of Photosynthesis revision video from scienceworksheets.co.uk. Please like, subscribe and comment to support the channel. The aim is to investigate the effect of light intensity on the rate of photosynthesis using an aquatic organism such as pondweed, and we will measure the rate of photosynthesis using an aquatic plant, and we will explain the results using limiting factors. Key setup points: you cut the pondweed under the water. This prevents air bubbles blocking the xylem vessels. The pondweed is positioned with the cut end facing upwards, and the lamp is placed at different measured distances from the pondweed. For the method, you place the pondweed in a beaker containing sodium hydrogen carbonate solution. The sodium hydrogen carbonate will provide a source of carbon dioxide. You position the lamp 10 centimeters from the pondweed. Leave the setup for 2 minutes to allow the plant to acclimatize, and you count the number of bubbles of oxygen produced in 1 minute. You will repeat the count and then calculate a mean, and you move the lamp further away at regular intervals, repeating the measurements. The variables. For the independent variable, this will be light intensity, usually changed by altering the distance of the lamp to the pondweed. The dependent variable is the rate of photosynthesis, which is measured by either the number of oxygen bubbles produced per minute or the volume of oxygen produced. Control variables, these need to be kept constant, such as temperature, the carbon dioxide concentration, the same pondweed or the same length of pondweed, and the same colour of light or the same type of light. We have our graph here of our results in our data table, and as you can see, as the distance of the lamp from the pondweed increases, the mean number of bubbles per minute will decrease. As the distance between the lamp and the pondweed increases, the light intensity will decrease, and so that is why the number of bubbles per minute will also decrease. As the light intensity decreases, the rate of photosynthesis decreases, and there is less light energy available for photosynthesis. So this is why the mean number of bubbles per minute decrease. At low light intensity, light will be the limiting factor. At higher light intensities, the rate levels off because another factor will become limiting, such as carbon dioxide concentration or temperature. Limitations to the experiment include the bubble size varies, the temperature may increase due to the lamp emitting thermal energy. Possible improvements could be using a heat filter or water bath. See our diagram, where this has a water tank between the lamp and the pondweed to absorb the heat energy of the lamp. You could measure the volume of oxygen using a gas syringe instead of counting the bubbles, and use an LED lamp as it emits very little thermal energy. Thank you for watching. See our revision notes, the link is in the description.